Good morning. I'm Bob Matthews from the University of Massachusetts Medical School, and I'm most pleased to introduce Dr. Celia Schiffer, Professor of Biochemistry and Molecular Pharmacology, as a recipient of the 2020 William C. Rose Award for the American Society of Biochemistry and Molecular Biology. The Rose Award recognizes outstanding contributions in science and mentoring, and Celia is a stellar example of both. Celia received her undergraduate degree in physics from the University of Chicago before joining the graduate program in biophysics at UCSF. Her thesis work in structural biology was directed by Robert Stroud. While there, she also developed an interest in computational biology that led to a postdoctoral in Zurich with Wilfried von Gunstern, where she focused on the application of molecular dynamic simulations to biological systems. Returning to San Francisco, Celia enhanced her skills as an X-ray crystallographer during a second postdoc with Tony Kosikoff at Genentech. With skills in structural and computational biology in hand, she joined the faculty in biochemistry and molecular pharmacology and began her career at the interface of both disciplines. In our medical school environment, she chose to focus on the development of drug resistance in proteases from HIV, hepatitis C, influenza, and Zika viruses. Careful studies of protease drug complexes led the development of the substrate envelope complex hypothesis, postulating that drugs whose structures closely resemble that of natural substrates would be more likely to avoid resistance by mutations in protease. You will hear more about that today in her lecture. Combining structural analysis, molecular dynamics, and synthetic chemistry to test her ideas, Celia has built a powerhouse research program that attracted major funding, published numerous papers, and trained dozens of students and postdocs. As a result of these successes, she became the founding director of the Institute for Drug Resistance at our medical school, and she was named a fellow of the American Academy of Microbiology in 2015. As I mentioned earlier, Celia has also excelled in mentoring her students, postdocs, and her younger colleagues at our institution and elsewhere. She has a passionate interest in both their professional and personal development, seeing them as part of her larger family. Her impact is best described by those whose life she has touched, and I offer two examples in the following quotes. From a former graduate student, Celia's mentorship didn't end with my graduation. Throughout my postdoc, Celia was there for me in the same capacity she had as a graduate student. When I was having a difficult time as a postdoc, Celia was the first person I called and was there to listen and provide invaluable advice. She was there to discuss the science I was pursuing and provide some helpful tips when I ran into difficult experiments. In pursuing faculty positions, Celia never hesitated to provide the necessary letter, but went beyond that, practicing my talk on the phone with her and reviewing all of my application materials. She continues to this day to support me in my career and is still my biggest cheerleader. From a junior colleague in the department, personally, Celia was my mentor from the time that I joined the department until I was awarded tenure. She offered me support and guidance in all aspects of my career. Celia helped me with grant proposals, advised me as I learned to deal with conflicts in my laboratory, and helped me to navigate the challenging path that leads to tenure at the medical school. I'm extremely grateful for her mentorship, which has certainly contributed to my success. Finally, it will not surprise you to learn that Celia has been fully committed to diversity during her entire career. She has recruited many women with diverse backgrounds from all around the world, and half a dozen underrepresented minority students have won prestigious graduate fellowships from the NIH. With no further delay, I'm pleased to introduce Celia Schiffer as a 2020 Rose Award winner.
Hi, I'm Celia Schiffer. Thanks so much for being here today, and I hope you and yours are well during this most trying of years. I'm sorry we can't be together today. I'm going to tell you about constraining evolution and how to avoid drug resistance and some of the lessons we've learned from viruses. But first, I really want to thank everyone, and especially for Bob Matthews for the kind introduction, and all my mentors and colleagues who nominated me for this honor, for the ASBMB to make me the Rose Award, and to the generations of my laboratory and my family, my parents and my daughter. You're the last couple decades of my laboratory who I had the honor to work with and mentor, and I'll be telling you about some of their work over the next few minutes in my current lab as of April of this year. So with that, I want to give you a little bit more introduction about who Dr. Rose was, because he was really very remarkable. He was a biochemist and a nutritionist who spent most of his career at University of Illinois, Urbana. He was really into understanding quantitative amino acid metabolism, modifying the diets of rats and then graduate students. And he discovered, he was after an understanding of what amino acids are essential for growth. He actually discovered the last amino acid to be discovered, threonine, which I hadn't known before, and which was published in JBC in 1935. He was president of the predecessor of this society in the late 30s, early 40s, received the National Medal of Science in 1966. And this award was established in honor of his 90th birthday by his mentee. So what an amazing honor to have such an award. So with that, I'll go back to telling you about our science and thinking about how we constrain evolution to avoid drug resistance. Now, drug resistance impacts millions of people and costs billions of dollars. It occurs whenever we try to restrict a quickly evolving disease or system. So if you think of a patient who's not feeling very well and has an infection and takes some medicines and starts feeling better, but unfortunately, resistance starts happening and he starts feeling worse again. And this is basically that the medications are no longer effective. And it goes from viruses to bacterial infections to cancer and beyond. And it's also a problem in agriculture and herbicides and pesticides. So how can we be smarter about avoiding drug resistance? And I'm a structural biologist, so I'm thinking between structure-based drug design and evolution. How does drug resistance often occurs when there are mutations in the macromolecular drug target that allow it to retain function while no longer being efficiently inhibited by the drug. Part of this is realizing that disrupting the activity of the drug target is necessary, but it's not sufficient for developing a robust drug that avoids resistance. We really need to think about designing drugs to last. So if we think about a normal disease progression, and most of the diseases that most of the drug targets that we go after are enzymes, and they interact with a particular substrate to make a functional complex. And then the disease progresses and you have an unhappy patient. When we do standard drug design approaches, whether it's high throughput screening or structure-based drug design, we screen against a target. If we're lucky, we get a hit. If we're really lucky, we can get it to be bioavailable and all those good things. But then if it's a quickly evolving disease, resistance can occur. And it occurs in such a way, either due to, in a virus, maybe the infidelity of a polymerase, or in a cancer cell because a base editor, such as ApoBec3, starts making mistakes. And then the drug doesn't bind so well anymore. It doesn't fit quite as well. And the enzyme's able to continue to recognize the substrate and keep going. So we've developed a new paradigm to avoid drug resistance where we really pay attention to the enzyme substrate complex. And if the enzyme interacts with more than one substrate, so much the better, because then it's evolutionarily constrained to avoid drug resistance. And so we design inhibitors that fit within the substrate envelope because a mutation impacting them will simultaneously impact the recognition and processing of the substrates. So these inhibitors still have to be potent and bioavailable and all those good things, but a mutation, but they will be able to be potent even in the face of mutations or other polymorphisms that might exist. So if we compare our paradigm with more traditional methods, there are lots of opportunities for resistance to occur, and we're trying to decrease the probability of resistance occurring with the idea that disrupting a drug target's activity is necessary but not sufficient for developing a robust drug that avoids resistance. Another way of thinking about this is that resistance occurs when the biological function of the target is favored over inhibition. So it's really not just the target, but the environment that exists, the structure, the dynamics of the free and the bound form. So think of your freshman chemistry class and free energy of binding. I really think about this in terms of a multi-dimensional space where you have the protein structure and its dynamics, the inhibitor structure and its potency, and the sequence diversity of the target itself. And we're trying to parse this multi-dimensional space to best inhibit that. And to do so, we really need to integrate many different techniques. My laboratory does a lot of crystallography. We do a lot of work on viral proteases that I'm going to touch on today, but also on the ApoBec3s, which our cytidine deaminases. We use a lot of enzyme assays. And we've also developed a methodology of parallel molecular dynamic simulations that allows us to distinguish differential behaviors between different similar systems. And so this is key, the incorporation of all these methods together. So today I'll tell you about two short stories. One discussing the specificity of ApoBecs. ApoBec3s are part of our innate immune system, actually help us to try to fight off viral infection, but they may also contribute to resistance. And a technology that we're developing to incorporate machine learning to decide for remote mechanisms of resistance, remote from the active. Let's start with the first story about base editors, the ApoBec3. Now, ApoBec3s are fascinating enzymes. They deaminate cytidines, single-stranded DNA, and RNA. Their signatures are showing up in all sorts of cancers, in breast, bladder, lung cancer. And this diversity is likely to facilitate resistance, because the more heterogeneous of cancer, the more likely it's not going to respond to therapy. Interestingly, these enzymes are only in placental mammals, and they're some of the fastest evolving enzymes in the human genome. A3H, for instance, there's 7 different haplotypes in human beings around the world. So it's really a quickly evolving system. We're studying the ApoBec3 family, and specifically the ones with Z1 domain. These ApoBecs are quite heterogeneous, even though they're on the same chromosome. Some are single domain, some are double domain. The double domain only have one catalytically active domain that we'll be focusing on today, where I'll tell you about our work on A3A, A3, and A3G. So a few years ago, we solved the crystal structure of A3A bound to single-stranded DNA, and it wraps around its substrate, recognizing TC motif in the active site, and there's a whole network of hydrogen bonds. It does so by wrapping around this histidine 29, which is a gatekeeper residue, and sort of wraps around in this sort of U-shaped conformation. And so we were really interested in how this structure conferred specificity. So we saw this U-shape to recognize the TC sequence. Our colleagues, Hiroshi Matsuya's group, solved crystal structure of A3G with single-stranded DNA. And A3G's recognition sequence is more of a CC sequence going into the active site. And so what we were interested when we looked at this crystal structure is that the network of hydrogen bonds is quite different. It no longer wraps around the histidine, but forms a more extended conformation, sort of cascading down the surface of the protein. And so we were really intrigued by the question of these different conformations that the ApoBecs are recognizing their single-stranded DNA, and we're curious about whether that was giving specificity to these enzymes. So in this case, the ApoBec3 G has a more extended conformation. So what are the structural mechanisms for differential specificity? What accounts for this difference? Is it the shape of the active site? Is it the sequence of the DNA? That's what we were trying to puzzle out. And it even becomes a little bit more complex than that, because the sequences, when we align them, there's a lot of homology between different ApoBecs. So here's A3, all of the Z1s, A3A, A3B, and A3G. But how potently they bind their DNA substrate sequences when in the inactive form varies tremendously, binding from 85 nanomolar to 160 micromolar. There are differences in these loops that are highlighted here in orange, and I'll show you them in a moment. But what really accounts for these differences in binding affinities and differences in specificity? So if we look at the structure, there are these 4 loops that surround the active site. There's loop 1, which has a lot of heterogeneity between the different ApoBec3s. And has this gatekeeper histidine from A3A, but there's a lot of variation. There's loop 3, which also has a lot of variation, but it's a little further from the active site. And loops 5 and 7, which are very highly homologous. And so we suspect are less important for specificity. But there's also a conundrum. A3A can also recognize a CC substrate. So even though it prefers TC with the 85 nanomolar, a CC substrate can still bind only about threefold weaker. So we were curious in understanding how these different enzymes recognize the different substrates. So we turned to molecular dynamics, and Sharon Ho did a series of molecular dynamics simulations and all the different structures with the different substrate sequences in the linear form, in the more wrapped around form. And you can see this is the A3A, how the DNA stays wrapped around the histidine. It's very stable in this simulation. And if we look at this more carefully and look at time shots across that, this conformation, this wrap stays stably around the enzyme, forming sort of a U-shaped shape. And if we go from the beginning of the simulation to the end, it's a very stable molecular dynamics simulation. But Sharon took it one step further and changed the sequence from TC to CC and did the same simulation. And this simulation, no matter how she started it, would settle out into the more extended conformation. So what was fascinating is that the difference that we could only discern from the dynamics was able to see the differential specificity from TC to CC was not just the protein structure, but it was the DNA substrate of how it bound to the enzyme. Now, if we drill down a little bit closer, you can see the details of this, how the T makes a nice network of hydrogen bonds with aspartic acid 31, including coordinating water, where the CC coordinates the aspartic acid in quite a different form and then starts packing against bases going further down the single-stranded DNA. So substrate specificity for ApoBec3A, you really need to take a multidisciplinary approach to understand the interdependence between substrate specificity and the conformation of the DNA. The TC motif wraps around the gatekeeper residue of the enzyme, where the CC motif, it binds in a much more extended conformation. And we can get that from simulation and are currently continuing to test this model experimentally. But let me move on to my second story. And this is one of deciphering the mechanisms of resistance. Now, HIV is an ideal system to understand mechanisms of resistance because it evolves so quickly. Just to give you a feel for how quickly HIV evolves, this is the genetic diversity of the global pandemic of influenza in 1996 compared to the diversity in a single person who had been infected with HIV untreated for a year. But that diversity is about the same. If you compare the diversity to within the country of Congo before a therapy was available, there's much more diversity. This fast evolution allows us to really study the mechanisms of resistance, that coupled with the existence of extremely potent inhibitors, my favorite ones being the HIV protease inhibitors, in particular darunavir. HIV protease is a viral protease that recognizes and cleaves a whole series of different substrates in the GAG and polyprotein, which permit viral maturation. And many years ago now, we discovered that what this enzyme recognizes in terms of substrate is not a particular amino acid sequence, but it's a conserved three-dimensional shape. And this was the basis of our substrate envelope hypothesis when we discovered that where primary resistance in the active site occurred was right where the inhibitors protruded out from that substrate envelope and contacted the enzyme. So we were interested in understanding how these primary resistant mutations work, and many of these primary mutations are mutations like isoleucine 84 going to a valine, isoleucine 50 going to a valine. Both of those confer high levels of multi-drug resistance. But in highly resistant patients, there can be over 25 mutations in the 99 amino acid sequence that confer resistance. And nevertheless, the protease is still functional. So darunavir, we took darunavir and we made a whole series of analogs of darunavir to understand the levels of resistance. And all these inhibitors fit within the substrate envelope. And our colleagues, Ron Swanstrom's lab at UNC Chapel Hill, took our inhibitors and did viral passaging to get high levels of resistance. And they passaged the viruses with suboptimal amounts of inhibitors to understand the resistant pathways that were occurring, taking our picomolar inhibitors and selecting for resistance that could tolerate 10 micromoles of inhibitors in viral culture. So these are really highly resistant variants. And with deep sequencing technology, we could follow the pathways of resistance, the mutations that arise as the virus becomes more and more resistant. And interestingly, even the same drug could have different pathways. So here's an example of this, one which started out with the active site mutation I84B, and then added more and more mutations to confer resistance, and the other with the I50B. But both of these active site mutations occurred early in the selection of resistance, and the other mutations were added on that I'll show you in a moment are outside the active site and confer high levels of micromolar resistance. So to characterize this more broadly, we made enzymes to look at the different variations within the protease itself. And on average, there were 10 mutations per viral protease. And in fact, when we measured the KIs, we lost a huge amounts of potency. These inhibitors bound with less than 5 picomolar potency to wild type, and now they were close to 1 micromolar, especially if you look at the stark blue and red, the levels of resistance to the protease inhibitors were extremely high. So if we look at these red and blue, here again are 2 different pathways to get high level of resistance to all of our inhibitors that we tested. So, you know, it's not 1 or 2 mutations, but it's a whole family of mutations that are causing high level of resistance. So let me walk you through how we got this high level of resistance. And this was our most resistant variant we've seen to date. It has about almost 1 micromolar Ki. And so if we look at, you know, the wild type, as I told you, finds about 5 picomolar. With 1 mutation in the active site, we lose about 5-fold resistance. 2 mutations in the active site, it's about 50-fold resistance. 2 more mutations now on the back side of the flaps of the enzyme, we're at 80-fold resistance. Another 4 mutations in the core of the enzyme, and we're up to 2,500-fold resistance. 2 more mutations outside the active site, and we're at 30,000-fold resistance. So these aren't active site changes that are causing resistance, but high levels of resistance outside the active site. And the final mutation that got us up to 150,000-fold resistance is, again, outside the active site on the back of the flap. So we have now this beautiful panel of resistant variants. And the question is, what are the molecular mechanisms? And so we looked at this aminohenase and then Forney Lockbound solved crystal structures of all these variants, but the crystal structures of all these variants didn't show us the mechanisms of resistance. They look very similar. So then again, we had to turn to parallel molecular dynamics. And so when we did the molecular dynamic simulations of darunavir bound to the wild type form, the drug stays pretty much put. It's happily in the active site. But when we start looking at the most resistant variants, and we looked at everything in between as well, you can see darunavir is pulling out of the active site. The protein's not happy. It's really pulling away. So there's lots of things going on in these simulations. So how do we look at them? We started looking at things like correlation coefficients. How well does the drug fit with the enzyme as the simulation goes on? And you can see the wild type's very correlated, and this correlation gets less and less with high levels of resistance. We can look at things like RMSF fluctuations of the drug, and with resistance, we're getting higher and higher levels of fluctuations with the drug and the enzyme with resistance. But to really understand what the molecular mechanisms of resistance were, we had to take this one step further. And this is where we pulled in machine learning. So we did parallel molecular dynamics simulations where we have many simulations of fairly short 100 nanosecond simulations, pull out parameters of van der Waals, hydrogen bond, water structure, and so forth, and then apply a regression analysis machine learning algorithm to interpret the results. We used about 30 different variants, ranging in binding affinities picomolar to micromolar, and mutations from 0 to almost 25 mutations. And so all these things were fed into the molecular dynamics simulation. We pulled out the features, the van der Waals torsions, and so forth, did the regression analysis. And then most importantly, we got coefficients out that allow us to have interpretable results. So the simulations with machine learning give us interpretable results. And these features give us very specific interactions that are of the thousands of interactions, only a small handful really describe the data set. So we're able to look at that small handful in detail. And when we map them onto the structure, they bisect the active site, but not through the dimer symmetry of the enzyme, but diagonally through with van der Waals contacts, with hydrogen bonds to the drug, and so forth. And so if we can, and with these small numbers of features, we can fit the data extremely well. So we had split the data between the ones that we used to develop the model, the training set of 18 complexes, and another 10 that were test set. And you can see that the training set in blue fits to the experimental data from picomolar to micromolar within a kilocalorie of binding affinity. And even the test set that were not included in the model, they fit to the experimental data within a kilocalorie for the most part. So this is extremely powerful of this range to understand what are the contributions to resistance. And just for comparison, here it's compared to MMGBSA snapshots through the trajectory where it's much more of a scatter plot. So the machine learning is really turning out to be a powerful technique. And beyond that, we can really look at the mechanism. So if we look at the top 4 interactions, you can see, satisfactorily, the indicators by which resistance occurs, the flaps are opening up. So between 84 and 50, that whether it's for mutations or van der Waals pulling away, goes almost linearly with resistance. The loss of hydrogen bonding to the drug also goes almost linearly with resistance. Both of those are very reassuring, but also more remote changes, a relaxation of the packing of remote residues, even way out at the edge of the core. There, as the enzyme opens up, you're getting that correlates with resistance in a very linear fashion. So these are bellwethers of resistance that we can only get by combining crystallography, computational chemistry, and the enzymatic assays. So now I hope I've given you a feel for the landscape of resistance, how multidimensional it is, and how we combine protein structure, dynamics, with inhibitor binding and sequence diversity. And I think it's only through that and by restricting enzymes like ApoBec3 that we can take a preemptive role to avoiding drug resistance. And these lessons learned from quickly evolving diseases are not limited to viruses, but are just the same in oncology and other infectious diseases, and as well as in agriculture. So these last lessons translate. With that, I really want to thank my colleagues, my collaborators, the Society again for the honor of the Rose Award. And I thank you for your attention and hope you and yours are well during this most challenging of years. Thanks again.